Your heart. It's gonna beat somewhere around 3 billion times over your lifetime. But what if I told you that every single one of those beats is governed by a tiny protein, a little titan, so small you can't see it, but so powerful it literally holds the key to life itself? Today, we're gonna go find it. 3 billion beats. Just let that number sink in. It's an absolutely incredible engine. But what happens if that perfectly tuned machine can be completely derailed by one tiny mistake? A single typo in its genetic instruction manual. Well, the results can be devastating, and it all comes down to the tiny protein we're about to meet. So get ready. We are going on a journey, and we're going to zoom way in. We'll start with the whole heart, then dive down into its tissues, past the individual cells, and get right to the molecular machinery that makes it all tick, the proteins, and the genes that build them. Okay, our first stop is at the tissue level. Your heart is made of this really unique material called cardiac muscle. And trust me, it's an engine unlike any other in your body. So what makes this tissue so special? Well, for one, it's only found in the heart. Its fibers are branched, all interconnected. And these connections, they're called intercalated discs, and they are absolutely key. They make sure that when one fiber gets the signal to contract, that signal spreads like wildfire, creating a single, powerful, synchronized beat. And the best part? It's all completely involuntary. Thank goodness we don't have to think about doing that 3 billion times. Now let's zoom in even further, because inside every single one of those muscle fibers is this beautiful, intricate molecular dance that creates the beat itself. It's called the sliding filament model. The stage for this dance is a microscopic structure called the sarcomere. The best way to think about it is like the fundamental engine block of the muscle. You've got millions of these things lined up, end to end, and they are packed with the protein machinery that does all the heavy lifting. And on this stage, we have two star performers, actin and myosin. Just picture actin as these long, thin ropes anchored at either end of the sarcomere. Then you have the myosin filaments. They're thicker, and they're covered in hundreds of tiny little heads that act just like oars on a boat. The entire game here is about those oars grabbing onto the ropes and pulling everything closer together. You know, our understanding of this whole dance didn't just appear overnight. Scientists back in the 1800s saw hints of it under their early microscopes. But the huge breakthrough came in 1954 when two scientists, both named Huxley but famously not related, figured out that these filaments don't actually shrink. They slide past one another. This was a game changer. And that discovery paved the way for another massive leap in 2003, finally linking a specific gene called PLN directly to heart disease. And this is exactly why that history is so important. As this quote gets at, it's that deep, fundamental knowledge that fuels today's medical breakthroughs. Understanding how filaments slide past each other in 1954 is precisely why scientists today can develop incredibly targeted gene therapies for tomorrow. It all connects. So here's how that rowing motion actually works. It's called the power stroke. First, a little bit of energy from a molecule called ATP cocks that myosin ore back into a high-energy position. Then it binds to the actin rope. Next up is the power stroke itself. It pulls that rope, shortening the whole unit. Now here's the crucial part, and it's a bit counterintuitive. A new molecule of ATP has to come in just to make the oar let go of the rope. That's what gets it ready for the next stroke. But hold on, these oars aren't just rowing constantly, right? Something has to tell them when to start. And that signal? Well, that's the master switch for all muscle contraction, calcium. The spots on the actin rope where the oars need to grab on are normally blocked. So what on earth gives those myosin heads the green light to grab on and start pulling? The answer lies with a couple of gatekeeper proteins. There's this long ribbon called tropomyosin that physically covers up the spots where myosin wants to bind. But when the electrical signal for a heartbeat arrives, calcium ions flood the cell. This calcium then grabs onto another protein, troponin, which acts like a handle, yanking that tropomyosin ribbon out of the way. And just like that, the binding sites are exposed and the power stroke begins. Contraction happens. Okay, so if calcium is the on switch, the accelerator, how does the muscle turn off and actually relax? This is where we finally meet the tiny titan of our story, a protein called phospholambin, or PLN for short. It's not the gas pedal, it's the heart's brake pedal. And just like every other protein in your body, PLN starts as a set of instructions coded in a gene. Those instructions get read, the protein gets built, and then it's shipped to a very specific spot, the wall of the cell's internal calcium storage tank, a place called the sarcoplasmic reticulum. So what's its job there? PLN has one main purpose, to control a pump called SERCA2A. And that pump's job is to suck all of that free-floating calcium back into the storage tank. When the calcium is gone, the muscle relaxes. So think about it. PLN controls relaxation, which is the crucial second half of every single heartbeat. That brake pedal analogy is honestly perfect. See, normally, PLN is on. It's physically pressing down on that circa pump, slowing it down. But when you need your heart to beat faster, like when you exercise, your body sends a signal, a process called phosphorylation. This signal basically forces PLN to lift its foot off the brake. The circa pump then goes into overdrive, clearing calcium super fast so the heart can relax quickly and get ready for the next powerful beat. So, what happens when that brake pedal is faulty right from the start? This is where a single typo in the PLN gene can lead to incredibly serious, life-threatening heart conditions called cardiomyopathies. Over 1,500. That's how many patients have been identified around the world with just one specific typo in the PLN gene. It's known as R14del. And honestly, that number is probably a huge underestimate. And this isn't just a string of letters in a scientific database. The story of this variant actually began with its discovery in a large family in Greece. And from that one finding, a global patient community has grown, including a foundation, all driven by the very people affected, all of them pushing science forward to find a cure. It's amazing. This just goes to show you how ridiculously precise our genetic code has to be. That R14del variant, a tiny, tiny deletion, causes the heart walls to thin out and weaken. But a different typo in the very same gene can cause the exact opposite problem, making the heart muscle grow dangerously thick. Two different typos, two completely different life-threatening diseases, all from one tiny gene. But here's where the story turns towards hope, because we understand this whole mechanism so deeply. From the sliding filaments all the way to this tiny brake pedal, scientists are now designing therapies to fix the faulty code itself. And the approaches they're taking are just incredible. We're talking about gene therapy, where you can deliver a healthy copy of the PLN gene. We're talking about gene editing with tools like CRISPR to go in and literally correct the typo in a patient's own DNA. Other strategies are trying to block the bad instructions from ever being read, or using drugs to help the faulty protein just work a little bit better. This is the absolute frontier of precision medicine. Which leaves us with this powerful final thought. For centuries, all we could do was treat the symptoms of heart disease. But now, with our growing ability to both read and rewrite the very code of life itself, are we on the verge of finally being able to fix the heart's smallest, yet most devastating typos? The race is definitely on.